Today we're going to talk about the cell cycle. And we're going to intro a good bit of material that we'll go into in more detail and repeat later on. But in general, the cell cycle is what we need for a cell to end up with two new cells. So a single cell, what it does, a single cell, essentially it copies its DNA and then ends up with two identical cells at the end that look very similar, almost an exact copy of what it started out with. And it needs to have the exact same DNA. So at the end, when there's two of these cells do not exist anymore. It's not like we're used to having, a parent and a daughter would both exist at the same time. This cell copies its DNA, which turns into this cell, and then this cell splits down the middle and becomes these two cells. And the first process we're going to talk about with this concerning this is PMAT. So we remember it by saying PMAT: prophase, metaphase, anaphase, telophase, and the acronym there is PMAT. And I'm sure you can go online and find different ways of remembering that, but PMAT is how I remember PMAT. And so very quickly, prophase, metaphase, anaphase, and telophase. Prophase, you've got a nucleus that, the DNA is typically in the nucleus, but in this case, the DNA can't get out of the nucleus, so it can't fit through the walls of the nucleus, it can't fit through those pores that we've talked about before, so the nucleus itself, the wall, has to break down. And then the chromosomes that are in there, they line up down the middle, which is how we remember metaphase, and then those chromosomes are being pulled apart in anaphase, and then in telophase, and then in anaphase, the cell is getting longer and longer, it's starting to pull apart, it's starting to be ripped apart. In telophase, you see it is very clearly being ripped apart in two different sides, and the nucleus for each side is now reforming. And then we call this little dip here, the belt around the figure eight, is going to be called a cleavage furrow. This is called telophase, and there's ways we can remember. Pro kind of sounds like pre, like it's the thing before, or the proto, it's the first phase. Metaphase, the things line up in the middle. Anaphase, we see these little lines. They look, to me, if you were to look closely, they look kind of like little half-drawn A's. So the A with that little bridge, and they're being pulled apart or away from each other. That's anaphase. And telophase, I remember that because a telephone is something you listen to sound from a distance. So if you call a friend of yours and you start talking to them, 20 minutes later, if you looked beside you and saw your friend in the seat next to you or seat beside you on a road trip, that would be kind of an odd situation. You talk to people when they're at a distance from you on the phone. You don't talk to them when they're right beside you typically. So telophase, meaning that these nuclei are actually very far apart at this point. And so prophase, metaphase, anaphase, telophase are dealing with nuclear division, and we see that down here. We'll talk about this whole pie chart here in a second, but nuclear division is what this is called, and it's called that because the nucleus is dividing. If you'll notice, the last part of telophase, the cell is still one cell. It is not two cells. That'll be the next step. So prophase, metaphase, anaphase, and telophase is the division of the nucleus. And this can be called, typically we say PMAT. When we see PMAT, you'll think of mitosis. We'll also talk about a process called meiosis, where this process happens twice. All right, so down here to this chart, you may go ahead and draw a pie chart. And the proportions, the size of each of these little sections is not hugely important. If it was real, if it was more realistic on, this section being realistic, it would actually look something like this, because this first phase that we're going to talk about, these three sections, but the cell spends about 90% of its life in this phase. These phases over here, it looks like it takes up a good chunk, but in reality, it's just a little sliver of the pie. But we draw it like this so we can write in it. It'd be difficult to write all this stuff in a small little sliver. So let's start with the first stage we'll talk about at least. We tend to start right here in G1. Remember, this is a cycle, and a cell is going through this over and over and over and over and over. So the first step that we were going to start with is going to be called G1, gap 1. I think about growth. It helps me remember that this is a growth stage. So the cell is growing. We'll see in a minute when we get all the way around here this cell is just a newborn baby cell, so it needs to grow. So it's going to grow and grow and grow, and then it gets to S phase. This S phase is the synthesis stage. It's going to be where copying happens. Another word that we use, you'll see a lot, is replication. So replication of DNA. If you'll notice, there's a chromosome in there, and then after synthesis happens, we're going to see double the amount of chromosomes inside the nucleus. And then after that, we will see G2, which is where the cell is going to continue to grow. You might see organelles being copied as well, but it's a growth stage. So we got G1, S, and G2. Those are all part of interphase. And interphase, again, is where 90% of the life of the cell is happening in interphase, these three. The cell does its job during these phases. So if it's a muscle cell, it's constantly pulling and relaxing and pulling and relaxing during this stage. It grows and prepares to divide. That's what's happening. It's growing and preparing for the division, which is happening in these two over here, or 5 over here. So what does the cell result in? It results in, eventually, over here, we'll talk about this, it's going to result, you know what, let's pause that. Let's come over here. So it grows, it copies the DNA inside, and then it grows some more. In prophase is when these blue sections, this is, in this case, we're going to talk about mitosis. This is the nuclear division. So we got prophase, we've got metaphase. Metaphase is, again, where it lines up in the middle. Anaphase, where they're being pulled apart. They look like little A's. And then telophase, where they're separated really far. These nuclei, the nucleus in each cell is forming again. And it's, excuse me, and it's about to divide. And the next step that we've not talked about is cytokinesis. It's supposed to be one word, but I couldn't fit it in there, so cytokinesis. This is when the cytoplasm splits into 2 different cells. The cell itself, the cytoplasm splits into 2 separate cells. And these cells that are formed, if I drew them up here, would look identical to the parent. So the parent starts here, grows, copies, grows some more, goes through the nuclear division, and then it goes through the cell division. And then now there's 2 cells that are identical to the original. And so what does the cell result in? It results in 2 identical cells, and it has the same DNA. Now that's the goal. Sometimes, we'll talk about this later, that the DNA is not exactly the same. There's some issues that go on that may lead to some differences down the road. So the cell cycle steps are, again, G1, S, G2. We've got nuclear division, and then we've got cytokinesis. You can see those right here. Why does the cell need to go through G1 and G2? And it's to make sure that they're big enough, they're large enough to make 2 separate cells and survive. So the reason you don't see, an acorn having fruit on it, it'd be weird if an acorn, which is a baby oak tree, if an acorn had fruit coming off of it, that would be odd, because that acorn has not grown and developed enough to reproduce. And that's what's happening during this G1 and G2 phase. And then why does S phase need to occur? Well, it needs to occur because, again, at the end, we want DNA that's going to be the same amount of DNA as what we started with. And so to do that, we need to double it, because so we need to multiply times 2 so that we can divide into 2.